Greetings. Would like to welcome everyone. My name's Jordan Orange. It's a real privilege to be able to introduce the second Jeffrey Modell Foundation Speaker Series. It's also wonderful to know that so many extraordinary immunologists and dear colleagues from all over the globe are with us to learn together, to grow together, to strive to be able to do more within immunology and within our field. So grateful to the Jeffrey Modell Foundation for making this and so many other things possible. And this ongoing series really will serve as a global convening and with regularity. And so grateful to be with everyone today. It's a privilege to be able to introduce Dr. Don Kohn. Um, he's, I'd say such a inspiration to so many of us as, he's really worked within our field to push the bounds of what we are able to do and even conceive for our patients. He's long been at the University of California, Los Angeles, and is affiliated with many of the extraordinary institutes there, um, but has spent a lot of his career in California and associated with many of the different institutions within the system. Of course, he's best known to all of us for his pioneering work in gene therapy. And of course, his pioneering work in gene therapy for SCID, for ADA SCID, for innovating with regards to techniques. But to take it a bit further, Don is really just so passionate about what brings us together, so passionate about bringing answers to our patients, bringing answers that we can all utilize. And I have to say, just the thoughtful responses to emails that we send, and I'm sure many of you share that experience. I don't know how he does it all and maintains such a extraordinary output through his science, but so grateful that he's with us today to share some of where his work is and some of where our field is going. So I love the title that he's chosen, Gene Therapy for Inborn Errors of Immunity. It's taken us so long because we've got so far to come. And so, without further ado, I'll pass to Dr. Kohn, and then we'll jump into a question and answer session. And thanks to those who have submitted some questions in advance. So, Dr. Kohn, please.
Hello, I'm just waiting for sharing to be turned on. I think it is. Nope. Can you turn on the sharing for me, please?
Hi, Dr. Kohn. I'm here now. I'm going to enable the sharing. It's enabled now.
Okay, I hope you can all see my slide.
Yes.
Just waiting for a slideshow to open. Okay, well, hello. Good morning from Los Angeles. I'm really grateful to the JMF for inviting me to give this presentation. I hope it's useful. I'm going to try and cover the scope of the field of gene therapy for inborn errors of immunity, which is growing. And the title I came up with when I was asked for a title was "It's taken us so long because we got so far to go," which is from a Stevie Wonder song. But I think it really describes, we've made so much progress, as I think I'll try and show you, but we certainly have a long way to go to really have therapies for all patients. This is my conflict of interest. I'm going to talk about a lentiviral vector for ADA SCID that we developed along with colleagues at University College London, and that's now been licensed to a startup company, Rarity Public Benefit Corporation, and I'm an inventor of it, so I have an IP interest in it, but it won't influence what I say. I just wanted to start off just briefly, with a tribute to my mentor, Michael Blaze, whom many of you probably knew. I worked in Mike's lab as a fellow, and the major lesson I learned from Mike is that nice guys can be successful in science. And so I've always kind of kept that as trying to model Mike. And when I met with him after we decided I was coming to his lab, he asked me, "How would you like to work on a project on gene therapy for your fellowship?" Which I didn't realize, but would sort of define the next 40 years of my life. So thank you, Mike, for everything you've given to the field and to the patients and to me.
Hi, Dr. Kohn. Roger here. I just wanted to share, if you could share your presentation in slideshow mode.
Yes, sorry.
No worries.
How's that? Okay. So this is what I'll try and cover in the time I have. Give an introduction to using gene therapy for inborn errors of immunity. Talk about using lentiviral vectors, which has been the major modality to the present time in the last couple of decades. Then I'll talk about gene editing approaches that are being developed at a very rapid pace and are coming to the clinic. In fact, are in the clinic now. And then I'll end with just a few comments about the future, which I think is going to be in vivo gene therapy. So we know from the International Union of Immunological Scientists that there's more than 500 inborn errors of immunity. This is sort of a partial list broken up by categories of immune disease. And they can all sort of be lumped together under the rubric that they're due to mutations in genes needed for the immune system to develop or function. So we're mainly talking about monogenic disorders. There are certainly many complex multigenic immune deficiencies or inborn errors of immunity. But some of them can be overcome by giving the missing gene product. So for example, giving immunoglobulin replacement for XLA can make a huge difference in the quality of life and health of the patients. In some cases, suppressing the abnormal gene product, for example, a gain of function gene, can be useful using drugs like JAK inhibitors. But sort of the definitive therapy is providing a new source of normal cells by hematopoietic stem cell transplant. And that's first was done in the late 60s. And so generally we use healthy other donor, allogeneic donor stem cells. Gene therapy that I'll talk about today is correcting the patient's own or autologous stem cells. So that's gene therapy as it currently is being practiced. And so if we go back to that list of inborn errors, of the more than 500, 9 so far have been the subjects of clinical trials of gene therapy. So 4 forms of SCID, Wiskott-Aldrich syndrome that recently got approved as a therapy in the US and the EU. CGD, both X-linked and P47 autosomal recessive, LAD1, and then there's also been some work for HAE. Which I won't talk about because it's not hematopoietic stem cell based. And so we've got more than 490 remaining to be conquered by gene therapy if we can. And so just to start off quite basic, all these approaches that I'm going to talk about are involving hematopoietic stem cells. So this is just cartoons, the process of upper left, collecting the bone marrow from patients, bringing it to the laboratory, doing whatever processing is necessary. In our case, I'll talk about the processing for gene therapy. Then often the patient will get some conditioning or chemotherapy to make space in their marrow and or to immune suppress. And the cells are given, and actually getting the transplant's the easiest part because they're just given intravenously, and they'll home back to the bone marrow because in the bottom right you see that the bone marrow is actually part of the vascular system. So once they're put into the bloodstream, they will, when they pass through the marrow, they'll stick and start growing. And really the hard part for the patient is the upper right. We've just given them typically high-dose chemotherapy, and they may already be immune deficient. So it takes several weeks to months for the immune system to recover, during which time the patients need isolation, ongoing antibiotics, and other types of care. And actually, more recently, it's very rare now to do bone marrow harvest. In fact, it's getting to be a bit of a lost art. And mainly we use mobilized stem cells. And so the observation was that if you give someone GCSF, which is the growth factor we make to drive white cell production, high doses for about 5 days, stem cells leave the marrow, go into circulation, and you can collect them by leukapheresis. And typically now another drug, plerixafor, is added sort of on the day of collection that boosts the release of stem cells into the bloodstream more. So this patient is getting leukapheresis. There's two IVs coming in and out of them, taking their blood to a machine where there's a centrifuge that spins a gradient, and the density where the stem cells layer is sipped off. The rest of the cells go back to the patient. So in a continuous process over 3 or 4 hours, their blood volume will go through the machine 3 or 4 times. So it's very efficient at collecting whether stem cells have moved into the bloodstream. So on the bottom right, you see a leukapheresis unit pack, and that can then be used either directly or frozen. In our case, we'll talk about CD34 selecting it and gene modifying it. And so again, the vast majority of both allo transplants as well as gene therapy are now using mobilized peripheral blood rather than bone marrow. They're kind of the same cells, but this is a relatively easier process, I think, for the patient and yields more cells. And so allogeneic stem cell transplant can treat many of these diseases, and the main barrier are the immunologic ones. So in a healthy person with a normal immune system, they would typically reject the donor cells, even from an HLA-matched sibling, if they weren't first immune suppressed. But the main problem we deal with in bone marrow transplant is the flip side, where the donor's T cells react against the patient, and that's graft versus host disease. So clinically, managing allogeneic stem cell transplants is really modulating these immune responses until the immune system can sort of emerge and be protective. So the hypothesis for gene therapy with autologous stem cells is that the stem cells that are corrected with the normal gene, and we'll talk about ways to do that, can have the same sustained beneficial effects on blood cell production or function without those immune complications of allogeneic transplant, rejection, GVHD. And so this should, in theory, eliminate the risks of GVHD, which can really be a significant problem in 10 to 20% of allo recipients. And then also probably either you need no, or only in some cases, some immune suppression prior to the transplant. So and really, it is an easier transplant, although it still is a transplant. It still involves giving chemotherapy to make space. And as we've learned from early trials, when we don't give any conditioning or any chemotherapy to make space, the cells don't engraft well. So we really do need to open up the niche. Presumably the chemotherapy gets rid of some of the remaining stem cells in the marrow, so when you give back the corrected cells, they can home and engraft. So this is the cartoon slide of this process. So we start with the patient, we collect their stem cells, as I talked about. And then ex vivo in the laboratory, how we're currently doing it, we either add a gene with an integrating virus like a retrovirus or a lentivirus that will put the therapeutic gene into the chromosomes of the stem cell, where it can be passed on to all the daughter cells. Or now ex vivo, we can also edit the cells with things like CRISPR. I'll talk about that in sort of the latter part of the talk. Once those cells are prepared, nowadays we typically will cryopreserve them and do full testing to make sure they meet a whole list of predefined criteria for safety, for efficacy. Patient will get marrow conditioning. The cells are brought to bedside and thawed and infused directly. And so the disease that I'll talk about for the first part is severe combined immune deficiency, and probably many of the people listening know more than I do about this disease. But just to summarize it, SCID is considered the most severe human primary immune deficiency, where there's absent T and B cell function. NK function is present or not, depending on the genetic cause of SCID. And we now know more than 20 different genes, any one of which, if defective, can lead to SCID. And thanks to Jennifer Puck and other people with newborn screening, we now have a very good measure of the incidence, and it's about 1 in 58, 1 in 60,000 live births are affected by SCID. And so the SCID was uniformly fatal in infancy before treatments were developed, and the patients would have severe multiple infections, often chronic diarrhea, growth failure, and just very sick, and usually would die from the infections. There was one famous child who was maintained in a germ-free bubble for more than a decade because there wasn't an available donor. We don't do that anymore, but it kept him alive for more than a decade. And so, as I mentioned earlier, bone marrow transplant or stem cell transplant can be curative. And so, if there's an HLA matched sibling donor, the chances of success are 95% and up, really approaching 100%, limited only by patients who get severe infection before they get transplanted. The success rates for the 80% of patients that don't have a matched sibling have been lower. And that's using either a matched unrelated donor that someone found through the registry to share most of the HLA antigens, or now we've gotten much better at doing haploidentical, typically from a parent where there's a full haplotype mismatch. Current processes for either processing the stem cells or post-transplant cyclophosphamide, for example, can really reduce the risks of GVHD. And I'll show you an outcome slide in a minute. The form of SCID that I've worked on primarily is ADA deficiency. So adenosine deaminase, it's an enzyme of purine metabolism, and in its absence, patients can have SCID. And it seems it's about 10 to 15% of cases of human SCID. There's about 10 children born with ADA SCID in the US and Canada per year, roughly. And of the more than 20 different genotypes of SCID, ADA was the first one where the biochemical basis and genetic basis were determined and where the gene was cloned to allow gene therapy to be considered. And without going into the pathophysiology much, the ADA deficient SCID have profound panlymphopenia. So they're typically T minus, B minus, NK minus, although sometimes they have a few NK cells, from accumulated lymphotoxic adenine metabolites. So in the absence of ADA, the precursor adenine metabolites build up and can be toxic to lymphocytes. And so the therapeutic options, there's a unique one for this one form of SCID, and that ADA enzyme therapy was developed in the late 1980s, approved as an orphan drug in the US in 1990. And by giving injections once or twice a week ongoing, there's enough breakdown of the adenine metabolites that immunity can be partially restored. And it's really an outstanding bridge for newly diagnosed patients to get them in very good shape for a transplant, or patients have been kept on it for a few decades or more. Then there's the allotransplant options I talked about: matched sibling, matched family, matched unrelated, haplo. And then there's autologous stem cell transplant with gene therapy. So this is a slide from a paper now that I guess 13 years ago, reporting outcomes after allogeneic transplants for ADA SCID from 5 centers. I think it was 4 in the EU and 1 in the US, showing the survival based on the type of donor. And so at that time, matched sibling or matched family, there was a 90 to 95% chance of surviving and immune reconstitution. But then as you went to less well-matched donor, matched unrelated, mismatched unrelated, or haploidentical, the results were much worse. Fortunately, we're doing much better. These are mainly patients from 1990s to even some back to the 1980s. So transplant results really have improved a lot. And so the Primary Immune Deficiency Treatment Consortium, or PIDTC, did a large study reported a few years ago. I think it was 121 ADA SCID patients looking at their outcome after transplant. And so on the left is sort of going back to the beginning of the record for the PIDTC, where patients getting a transplant either with or without preceding enzyme replacement therapy had about an 80% chance of survival. At that time, the number of gene therapy patients wasn't large, but they were all surviving. But when looking at the more recent era from 2000 to 2017, there was not a significant difference, in fact, between allotransplants of all different types compared to gene therapy. And so, gene therapy has been limited in its availability, as I'll talk about. And so I think we can now talk to parents and patients more confidently that allotransplant results are really supportive of those approaches if need be. And so ADA SCID is historic in that it was the first human disease for which an approved gene therapy was attempted in 1990 by my mentor again, Mike Blaze. So they treated 2 patients where peripheral blood T cells were collected from patients on ADA enzymes. So they had enough T cells in circulation, they could collect them, transduce them with a gamma retroviral vector carrying a normal ADA gene, and reinfusing them in monthly cycles. And the patients had circulating T cells with the normal gene, which persisted, I'll show you. But it was thought that the frequency of the cells and their T cell repertoire diversity was likely too low to restore full immunity. So the patients remained on enzyme replacement therapy, I believe to the present time. But unfortunately, that may blunt the advantage to the corrected cell. So it was kind of done with one hand tied because it wasn't known if it would work. So there were really no clear-cut clinical benefits, and the outcomes are basically those of other patients on enzyme. Just a slide showing the technology. So showing with either a gamma retroviral vector or its sort of cousin, the lentiviral vectors, gene of interest, in this case would be ADA, is cloned into the virus plasmid. And when the virus is packaged, that gene is what the virus carries. So that genetic information, which is RNA in the virus particle, gets reverse transcribed, which is the retro step, into DNA that integrates and puts the gene into the chromosome. And both lentis and retros do the same thing. They have some differences that I'll talk about in a minute. And so Mike's group with French Anderson, Ken Culver, other people treated these two patients. And Linda Muhl, who was part of that group, published a paper about a decade later looking at the follow-up of the patients. And they did have some persistence of T cells expressing ADA with the gene present that could be detected by PCR, better in the first patient than in the second one. So this was really, a first step for mankind. This is the first human gene therapy, and although it wasn't, a clear-cut home run, it set the field. And I think, importantly, one of the direct outputs of that was the development of CAR T cells. So Steve Rosenberg at the NIH was working with Mike Blaze's group at the time and used the same gene transfer technology to introduce T cell receptors or chimeric antigen receptors into T cells. And then the field just really exploded. So now there are many, many people walking around having benefited from CAR T cells that I would argue came in the follow-up of the initial ADA patients. And so the advances in ADA SCID, one of the first major ones came out of the group at TGET in Milan, led by Alessandro Aiuti and colleagues. And they reported two patients with ADA SCID who were treated, by the way, I'm talking about retroviral-mediated ADA gene transfer to their bone marrow cells. And before they gave them back their cells, they gave them a low dose of the chemotherapy drug busulfan. So a full dose of busulfan might be 18 milligrams per kilo. So about a quarter of the amount you'd give, and no other drugs, so no cyclophosphamide, no fludarabine. And these patients both came from countries that couldn't afford ADA enzyme, which is expensive. So they weren't on it, so it wasn't there to blunt the selective advantage. And what they reported was that the immune system was restored in the patients and increased numbers and function of T and B cells. The patients remained well, multiple years after the treatment. And there was a much longer follow-up paper published in Blood in 2016. And this therapy has gone on to being approved in the EMA as Strimvelis. And so it's still available at TGET in Milan for patients with ADA SCID. About at the same time, there was another immune deficiency that was reported as responding to gene therapy, and that's X-linked SCID. So there are sort of ongoing trials in both Paris, Necker Hospital, and London, Great Ormond Street Hospital, treating patients with ADA SCID, with X-SCID, using the gamma retroviral vector shown here. So it carried a normal IL-2RG cDNA, and the graph is showing T cell counts in the patients. So they all start off at basically 0 T cells, as X-SCID patients would. And over the course of the first year or two, T cell numbers came up in them. So 18 out of the 20 patients had good T cell reconstitution. And this was actually reported before the ADA SCID. So this was the first reported successful human gene therapy. But then, as those of you around then know, really tragedy struck because 5 of these 20 initial patients developed T cell leukemia about 2 to 3 years later. In fact, one of them developed it 15 years after the therapy. And this is just some of their published figures about the leukemia. So you can see in panel A, the 2 dark lines are the 2 patients who will go on to get leukemia. At this point in the first year and a half, they just had good T cell counts. But in the upper right, you can see that about 30 to 35 months out, they had a really high spike in their lymphocyte counts up to 100,000. Panel C shows lymphoblasts, and panel D shows that by the time they presented with leukemia, they had picked up additional chromosomal abnormalities. And so the techniques for looking at where a vector has integrated into the chromosome in a pool of cells was just developing at the time, but they worked out that in at least 4 of the 5 patients, the vector in the clone that grew out was near the same gene, LMO2. This is a protein involved in stem cell growth. It's normally turned off in mature T cells, but it's known to be a proto-oncogene that is activated in some cases of T cell leukemia. And so we don't think the vector went there specifically. The vector went there in the semi-random pattern that it integrates, but when it goes there, that then can lead to this process. So that's kind of diagrammed here. So we start off with stem cells from a patient. The little red arrow is the vector. We treat the cells. It goes in each cell. It integrates at a different site in the chromosome, and that will restore the ability to make T lymphocytes from those cells. So that's the therapeutic benefit. But the integration site in the middle happens to be next to an oncogene, that it shows that clone expands and then picks up other mutation and goes on to form a frank leukemia or leukoproliferation. And so sort of that era now, around 2000, for the first time, robust immune reconstitution was achieved in clinical trials for ADA SCID, as I talk about XSCID, and also trials for X-linked CGD and Wiskott-Aldrich syndrome. But then some years after the treatments, there were high rates of genotoxicity where the gamma retroviral vector caused insertional oncogenesis, so inserting near an oncogene and turning it on. And so the count, I think, is 6 out of 20 of the XSCID patients for that cohort, at least 7 of 10 from a Wiskott trial, 3 of 3 in a CGD trial. And interestingly, with more than 70 ADA SCID patients getting retroviral vectors of a similar type, only 1 has been reported to develop a leukemia. So there's something about ADA that may make it less frequent. So in studying it, the lesson we learned that it's basically the enhancer elements in the virus that are causing this. So that's a period, during the Clinton campaign, they said it's the economy stupid. So here it's enhancer stupid. So that led to developing new generations of retro and lentiviral vectors. And so that's shown here. So the original vectors that were used in all the patients I've talked about to the present time were gamma retroviral vectors that typically infect mice. And at their ends, in their long terminal repeats at both ends, they have very strong enhancers and promoters. So I think part of the reason they were selected by scientists to use as vectors, those strong promoters led to high titers of the vector and strong expression of the transgene. But the risk was that enhancer, because enhancers can turn on genes upstream or downstream, forward or backwards, can turn on the promoter of a gene near it where it lands. And if that's a proto-oncogene, that can then lead to a clonal outgrowth. And so sort of a second generation of vectors were developed in the early 2000s where those enhancers are deleted. And in the plasmid form of the virus, it's deleted just from one LTR. When the virus goes through reverse transcription, it copies that deletion so that it's missing in both. So it's self-inactivating. So they're sometimes called SYN vectors. And so you design it then with an internal promoter, that's the PROM, to drive transcription of the transgene, but not have strong enhancer activities. So promoters like elongation factor, alpha-1, phosphoglycerate kinase are medium-strength promoters that don't have really much enhancer activity. So even if they happen to land next to a proto-oncogene, they're unlikely to turn it on. And so around that time, a little bit later, our group working with colleagues at University College London, Adrian Thrasher and Bobby Gaspard, developed one of these second generation lentiviral vectors. So the red X's at the ends indicate the enhancers are deleted. And by codon optimizing the cDNA and using this WPRE element that stabilizes the message, we got like 9 times more ADA per copy than if it was the wild type codons for ADA and it didn't have the WPRE element. So that a single copy of the vector makes 1 to 3x amount of ADA as a normal cell. So that's, we think, is beneficial. And so this will turn out to be a very good vector. It's small, it has a very high titer, it works very well. And so just to show you some data, this was a trial we did now, I guess, 8 to 10 years ago. The program was licensed to Orchard Therapeutics for a while. And together we did this trial where we, for the first time for the ADA, cryopreserve the cells. So I'll just walk you through this. So the patients get referred, we talk with the family. If they agree, we go through the informed consent process. Patient will get screening studies to make sure they're eligible. And it's basically, heart, major organ function. And if they are, then in this trial, they would get admitted for for a stem cell collection. And back then we were doing bone marrow harvest. Now we're using mobilized peripheral blood. On Tuesday morning, we'd collect their bone marrow, take it to the laboratory. It would get transduced with the lentiviral vector I showed you and frozen away, tested to make sure that it meets all the release criteria for sterility, mycoplasma, endotoxin, cell growth, ADA expression, et cetera. And when we know we have a good product, the patient comes back in and gets busulfan. And because we have a frozen product, we have enough time to give the first dose of busulfan, measure that patient's individual clearance from pharmacokinetic measurement, and adjust the second dose so they get an area under the curve that's relatively constant patient to patient, which is the way to dose it sort of consistently. And then on Friday, the cells are brought to the bedside from the freezer or laboratory, thawed and infused. And so we treated a total of 12 patients under this protocol. And so, as I said, we did all the work really in parallel with a group at University College London, GOSH, with Adrian Thrasher, Bobby Gaspar, and Claire Booth. So we published a paper jointly in 2021 where we had the results of 50 ADA SCID patients treated in these parallel trials. We had a common clinical protocol. The vector batches were made at Indiana University, and we split them between us. So we're using the exact same vector preps. So we used CD34 cells with the vector, the lower dose of busulfan that I talked about. And so the results were really quite good. So 100% of the patients were alive, and 96% of them were event-free survival, meaning it worked and they didn't need to have either a second transplant or go back on enzyme. The two that didn't work, one of them in London got a low cell dose. One of them that we treated, we don't know why she didn't engraft. But 48 of them did, and to this time still have good immunity. This is just one figure from that paper showing T cell counts. So when the patients came in at time 0, they were on enzyme, so they had some T cells. They got their stem cells collected, reinfused, and then you can see over the course of 1 to 2 years, their T cell counts came up into the normal range. The London group started a year before us, so that's why there's 3 years for the green line and 2 years for the blue. So we just published another paper late last year with a longer term follow-up. And so, again, Claire Booth from UCL, GOSH was the first author, I was last author, with the lentiviral vector we talked about. And now there were 62 subjects because another 12 had been treated by London sort of shortly after the last paper was closed. And so now we had 62 subjects who were 7 to 12 years after gene therapy. And basically all those who reconstituted initially had sustained immunity. There were no product or vector-related adverse events. This was actually a data cut from 2.5 years ago. So they're actually, I guess, 9 to 14 years out now. And there were no subsequent events. So there were 3 in this larger group that didn't engraft the cells early on, but the other 59 remain well and have sustained immunity. And the issue I mentioned about vector integration site, now there are very robust ways to amplify from all the integrated vectors in a population out into the surrounding cellular DNA, do next-gen sequencing, map that against the genome, and in a complex mixture of cells, map 10,000 different integration sites or more. And so that was done through the National Gene Vector Biorepository. And all of them showed kind of a multiclonal pattern of integration sites, no predominant site near an oncogene that had expanded. So again, it looks quite like a benign picture of integration. And so just to finish this story, so this is now clearly something that works well, and the challenge is no longer scientific; it's really commercial. So I mentioned that Orchard Therapeutics licensed this from UCLA and University College London in 2017, and in 2022 they decided to stop working on it and returned it to the university. So we got back the IND and all the intellectual property, and so we're trying to get it through an FDA biological license application for marketing authorization. You can't really get reimbursed by insurance companies for a drug unless it's FDA approved for the most part. And the main task that Orchard worked on that we are now addressing is we had done all the manufacturing of the vector and the cells in academic institutions, perfectly fine, clean, effective, but not done in a sort of a commercial pharmaceutical grade in terms of the facility, the quality systems, et cetera, which is required for an approved drug. And so we need to establish commercial grade manufacture of both the lentiviral vector and then the drug product, which are patient-specific lots of the modified stem cells. And so we've established a public benefit corporation called Rarity Public Benefit Corporation, which is a for-profit, but its charter says its goal is to make the drug available in a sustainable manner, not to maximize return on investment. And so they've licensed the IP from the universities. They've contracted with a commercial drug manufacturing organization to develop the commercial manufacturing needed. They've hired a commercial research organization to gather all the data, and we're just going to be presenting the U.S. data to the FDA, all the data from now I think it's 41 patients we've treated. And then ultimately they need to become a drug company and make it available once it's approved. And so they are doing that. I think they're making good progress. So I'm hopeful we will have this across the finish line within a couple of years. And the California Institute for Regenerative Medicine gave a funding for this work to move it forward. So they're currently the major funder. They funded the cryo trial that I showed you before, and they're funding this work. So then just to wrap up the lentiviral vector part of the story, so this is a list of all the diseases that I'm aware of that have had clinical trials of this approach: ex vivo transduction of hematopoietic stem cells with a lentiviral vector, conditioning, reinfusion, and all of these have shown clinical efficacy. The ones in yellow are FDA approved. The one in green is the one that's the Strimvelis approved in the EU. And these are the inborn errors of immunity. So again, the 4 different forms of SCID, WSCOT, both CGD, X-linked, and P47, and LAD have all had very excellent results. And several of these have been published, and a few of them are, like the P47 CGD is more recent, but the initial patients are looking good. So this approach does work, and if you make the vector right, it can be quite safe, as I showed you for the ADA. In the remaining time, let's talk about gene editing, which is really the up and coming. And so the basis of most current gene editing techniques are based on CRISPR. And just in case there's anyone out there who isn't aware of CRISPR, so CRISPR is a bacterial immune defense. So in this example, at the top, there's a bacteriophage inserting its genome into the bacterial cell. So bacteria have a defense system where we'll take some of that genome, cut it up, and put it into their memory bank. So they have this memory bank of pieces of viruses that have infected them before. That's the different colored squares. And these were found when people started doing high throughput sequencing of bacteria. They found these these clustered, regularly interspaced, short palindromic repeats. And it was determined that, in fact, those are expressing these little pieces of the viral RNA. And there's a protein called Cas9 in bacteria that can bind that RNA. And the next time that bacteriophage puts its DNA into the cell, in a complementary manner, that loop of RNA will bind to the DNA, and the Cas9 protein will cut up the DNA. So it's a double-stranded nuclease. And so it was really Jennifer Dowden and Emmanuel Charpentier who recognized that you could turn this into a gene editing system. If you can direct this protein to a specific site in the genome by matching RNA and cut it, you could do gene editing at that site. So this shows that Cas9 protein. And what you need to direct it to a specific genomic sequence is this short guide RNA that is complementary to your target system. And all you really need to target a sequence of the genome is, in the case of the standard Cas9, a proto-space or adjacent motif site that has a sequence anything GG, NGG. So in the sequence on the bottom, there are 3 NGGs, two A's and a C. You could make a CRISPR guide to each of those sites and test them to see which one most efficiently cuts the DNA at that site. So that led to sort of first and second generation editing techniques where you use CRISPR or something like that, TALENs, zinc finger nucleases, to make a cut at a specific site in the genome near your mutation. And then if you don't do anything else, the cell will most likely repair that double-stranded break by non-homologous end joining, which often adds or inserts some bases at the joint, so insertions and deletions, and that can be used to knock out genes. So Casgevy is a CRISPR that knocks out BCL11A, the repressor of fetal globin, to turn on fetal globin. That's beneficial for patients with both sickle cell or thalassemia. You can, in theory, knock out the HAV co-receptor CCR5 by that approach. Or you can add a homologous donor matching the cut site, but with the correct base, and that can then knock in the correct sequence. And we're actually doing a trial of that for sickle cell disease. Or in fact, you can get a whole donor that's flanked by the target site, homology arms, with a cDNA in the middle. And that can be used for many diseases, because now you can get a normal cDNA for a gene running off its normal promoter and get relatively physiologic expression of the gene, which probably for many inborn errors of immunity is important to have physiologically regulated expression right developmental stage of the cells. And so that's just cartooned here. So if there's a gene with a mutation in it, you can make a nuclease like a CRISPR to cut in the 5' untranslated region and make a donor molecule that's the whole cDNA. So it's all the exons put together with arms flanking it homologous to the cut site. And that can then integrate in there, and you could run this inserted copy off the promoter and ideally get physiologic expression of the gene. But we're moving on to now other sort of third generation editing techniques like base editing and prime editing. I'll talk about those just a little bit. And there's other things coming to put in large gene sequences like recombinases. So nucleases like CRISPR will make a cut in the DNA. Base editors, as cartoon here, is much more precise and can change a single letter. And if it's the letter that is the responsible mutation, you can revert it. And so that's shown here. So it uses the Cas9 protein to find the specific sequence in the same way. Cas9 has been disabled, so it either makes no cuts or only cuts one strand, so it's a nickase. And fused to that is a deaminase. So in this case, cytidine deaminase will turn cytosine into uracil. And so if you bound to the specific site by the Cas9 part, recognize it, and the deaminase changes that C to a U, when you fix the nick on the other strand, you'll put a G opposite the U, and you've converted the CT base pair, the CG base pair to an AT. And so cytosine base editors can change a GC to an AT. There are also adenine base editors that can change an AT to a GC. So we're basically doing transition mutations. We can't really officially do transversions at this time. So just to let me briefly tell a story, because this has been published. So Nikki Wright is in Alberta, Canada, has many patients who are Mennonites with a specific mutation in the CD3D gene involved in T cell receptor expression. So she asked us if we could make a vector for it. And in fact, I had a student in the lab at the time, Grace McCauley, who's now an MD PhD student at UCSD, who wanted to try base editing. And so the idea is on the left, you see the mutation. So it's a stop codon where the TA should really be a CG. And so she developed a base editor that bound to that site. The adenosine deaminase would deaminate the adenosine to an inosine, which is read like a G during repair of the nick on the other strand. So you converted the stop codon to the normal arginine codon. So Grace worked out all the activity. And this is just trying various versions of the editor, showing very high efficiency correction of the mutation. So then we worked with our colleague Gay Crooks. Nikki sent us a sample of bone marrow from a patient with CD3 delta SCID. Grace corrected it in vitro and then gave it to Gay Crooks's lab, who's developed an artificial thymic organoid that will take CD34 cells and turn them into mature lymphocytes. When we did that with the healthy donor control, we see the production of lymphocytes that are CD3 positive, TCR alpha beta positive. Nothing forms from the unedited patient cells. Those who were corrected are now making normal T cells, and we did extensive analysis of the T cells in the paper. It's actually my favorite paper for my whole career. And so now we're trying to focus this on XLA. So in XLA, there's more than 1,000 different mutations that have been described in patients. And by at least looking at their sequence, about 40% of them could be corrected by base editing. So here's an example of a patient who was referred to us who has this mutation where what should be a C is a T. So if you change the A opposite, you can change the AT back to a GC. And now this is getting to be relatively simple to initiate. And so a graduate student in my lab developed a base editor to, again, change the stop codon to the wild type, and tried various versions of the base editor, recognizing different PAMs, got high efficiency correction, and that restored calcium flux. And so, as I said, there's more than 1,000 different mutations, many of which can be reverted by a base editor. And the FDA has recently released a draft guideline about a platform approach where different mutations in the same gene can be treated under an IND. And so you need a different guide RNA to direct the editing machinery to the specific mutation. and you may need a different variant of the base editor to make sure it binds to that site. And so the main work is then to show for each successive mutation that you can get effective correction and that you don't have off-target activity that could modify a gene elsewhere. And again, the methods to do that now have become relatively routine and standard. So one of the challenges, the cost for high quality editing reagents are very high. Typically, they're made in relatively large lots, and under GMP grade, it can cost $500,000 just for the reagents. And if you're trying to treat multiple mutations, that's not possible. And so we need sort of a change in the manufacturing companies to make small lots of these reagents so that we can treat 5 patients with one mutation, 3 patients with another mutation in an affordable way. So the last method I want to talk about is prime editing. This is sort of the newest kid on the block. Again, it uses the same principles. A Cas9 protein is sort of the basis of it for finding the mutations with an RNA. But now the RNA has an addition on it. So fused to the Cas9 now is not an adenosine deaminase, but actually reverse transcriptase. And so once this nicks the other end, reverse transcriptase will copy the yellow RNA bases, will reverse transcribe them into DNA and insert them into the chromosome, which you can actually make small additions or other changes. So it's a very labile tool that you can really approach many types of mutations, not just single base pair changes. And so, in fact, it's now been in the clinic. This paper just came out from Prime Medicine, where they edited a P47 CGD mutation. So it turns out that most people that have autosomal recessive P47 form of CGD have a 2 nucleotide deletion. So the normal gene at the top, the middle gene is the wild type gene. It has a GT-GT sequence. That gene is flanked on both sides by pseudogenes where that's a single GT. And most patients with P47 CGD have lost the 2 nucleotides in the wild type gene. And so if you have a prime editor that can restore that GT, any one of the 6 genes could be corrected and lead to functional protein. And so what they reported in this paper was very high efficiency. So if we're looking down at the bottom in D and E, the DHR positive neutrophils are 70, 80% in the 2 patients in the time of observation. And so this was wonderful, but then the company decided to stop supporting this program because it was expensive relative to their planned in vivo trials. So we're hoping that some way this can be picked up and sustained because it's really quite efficient. So then the last comments are about ex vivo versus in vivo. So up until now, everything I've talked about is ex vivo, where we take the cells out of the body, modify them in the laboratory, give conditioning chemotherapy, and give them back. Where we'd like to go is to deliver the gene into the body, have it modify the stem cells in situ, and possibly that could avoid needing to do conditioning. So there's a variety of different gene delivery tools that are being studied for that task. So adenovirus, lipid nanoparticles, virus-like particles, all of which can be used to carry, for example, an editing payload and modify cells that take it up. So here's just an example of a lipid nanoparticle that's carrying an mRNA for a base editor, and a guide RNA. So those can then, in the target cell, the mRNA gets expressed translated into the protein for the base editor, and it can edit the genome in the cell. So there's one current that I'm aware of in vivo gene editing project ongoing. This is from a company called Insoma, where they're using an adenovirus, Ad35, that will infect human cells. And it carries a cassette shown on the right with both a normal CYBB gene for XCGD and also an MGMT chemotherapy resistance gene. That's flanked by sleeping beauty inverted terminal repeats. And a second virus giving at the same time expresses transiently sleeping beauty integrase. So it'll actually integrate that cassette into the genome. And so the approach is to mobilize the stem cells from the patient into the bloodstream, not collect them, but then give the virus IV, and hopefully it'll be more likely to collide with the stem cell because they're in circulation. Then after engraftment of those cells, chemotherapy agents are given that select for the cells got the resistance gene, so you can successfully increase the fraction of cells that have the corrective gene. And as far as I know, one patient so far has been enrolled and treated, and several centers are open. So that there will, if for people with XCGD, this is something to look into, although it's still quite experimental with just one patient treated. So then just to wrap up, so that's good and that's the progress. But as I said, we still have at least 490 more to work on. And one of the major imperatives is to get away from giving cytotoxic chemotherapy. It's not good for people. It makes them sick for several weeks from mucositis and other things in high doses. And so there's a number of approaches to try and make space without chemotherapy, many based on monoclonal antibodies to stem cell markers that can be given, can deplete some of the endogenous stem cells, and the cells can be given. There's a possibility that just mobilizing the stem cells and then giving the corrected stem cells opens up the niche. Or there are ways to try and confer an engraftment or proliferative advantage to the cells. Across the board, though, we need to improve the consistency of the engraftment, so there's certainly variability patient to patient. I talked about the new methods for gene editing that will allow us to have genes really be precisely expressed, and we need to reduce the cost for manufacturing. And the great hope is that in vivo will really go a long way to reducing the cost and the morbidity for patients. So it really comes down to, the techniques are good. They'll continue to get better. The challenge is access and availability. So the benefits of gene therapy have to justify the increased cost over allo products. So absence of GVHD, the ability sometimes to use less conditioning. The risks from gene manipulation must outweigh the risks of allo transplant, such as GVHD. I think they do, but we'll need more data to prove that. So these therapies have been provided at academic medical centers under research clinical trials. I showed you that long list of 18, 20 diseases. The complexity and the high cost for commercial grade manufacturing of vectors or editing reagents and autologous cell products is quite daunting. So only a small number have reached licensure in the US, the EU, UK, and it's really more we're limited by the financial issues of what's the business model for these rare diseases than their technical feasibility. And I will just stop there to thank, this is my lab group last summer, and this is people in my group that do all of our clinical gene editing and clinical trials. There are many, many collaborators without whom we would be still doing primitive things. And these are all of our funding. And of note, the JMF is supporting, was the initial support for the CD3 Delta work with Nikki Wright. And currently we have a specific defects grant to try and correct a mutation in the NEMO gene. And I will stop there. Thank you.
Thank you, Dr. Kohn, for a really comprehensive and informative overview, really from a perspective that you can provide. So we did have a bunch of questions come in live. We also had pre-submitted 90 questions. And so as people might imagine, that's impossible to accomplish over 7 minutes, but there's some themes that hopefully can be spoken to. As far as questions that have come in live, there was a couple of questions about XLA, which you went on to speak to, but also about what might be the given the number of patients with antibody deficiencies and certainly the longstanding understanding of BTK, what do you think is a realistic timeline and set of expectations for people around trying to cure humoral immune deficiency and particularly gamma globulinemia?
Yeah, good question. And we are hoping to be in the clinic in 1 to 2 years with the editing approach that I showed you. And so we now have sort of the top 5 most prevalent BTK mutations we've made that are base editable. We've made base editors and have laboratory data showing they work. We've put together a clinical protocol, so we're hoping to have a pre-IND meeting with FDA to propose to them how we would do this. And if they agree, then just add money and we'll do it. So we're really hoping to get to the clinic as soon as possible. And maybe 1 to 2, probably 2 years is
and just to follow that on, because with the regulatory guidances that have been emerging and some of the umbrella concepts that seem to be potentially available, does that give you optimism, or where would you like to see that go?
Yeah. So The test case is baby KJ, a little boy with a urea cycle defect born at CHOP who got in vivo gene therapy with a mutation-specific reagent that was developed in miraculous time. And I know I've heard the group from CHOP talk about their discussions with FDA, that FDA would consider that you could have in one IND multiple mutations of a single gene. So for XLA, all the different mutations that we'd want to treat could be under one IND. But if you want to treat a different immune deficiency with a different mutation, a different gene, you'd probably need a different IND. So you can have an umbrella protocol where the endpoints are the same, whether it's XLA or other, for example, humoral immune deficiencies. But it's still a fair amount of regulatory work to launch those. So they were told for the different genes involved in the urea cycle defect, but although they can have sort of a common protocol with common endpoints, they need an IND separately for each gene. I hope that will change once there's more experience with it. But even that, I think, was a great advance that will allow, again, for XLA, we don't need one IND for each of the 500 mutations we might want to treat. That one IND with just data on the specific reagents for each mutation may be enough to do.
Yeah, thanks for sharing that good news. And I think that is it's great to celebrate good news when when it's available. Having met baby KJ, that's certainly an inspiration. A question that probably is on many people's minds from Dr. Fernandez de Soto. Um, what continues to drive you? Why do you keep pushing forward, and what gives you the most hope?
That's a great question. I love what I do, and we just treated another group of ADA skid patients after it came back to us, and it's like performing a miracle. When you just go in the room, the patient gets this infusion of cells over a few minutes. It's not a lot of cells, it looks like, and you realize you may have just cured that patient of their disease. I mean, how often do we get an opportunity to do that? And so there's a number of diseases we're working on. I'd like to keep at it and find cures for more diseases. That's really my motivation. And it's fun, working in a lab with students and scientists in the lab. It's fun day to day doing it. And so I can't wait to get to work every day.
Yeah, I'm so glad to hear that, truly. From the many questions that have come in online, I think there's a theme around accessibility. There's a theme around equitable access, and I think in perhaps your last comments, you really do touch upon maybe some of the promise of LNP-based platforms and others to maybe sort of jump the whole innovation sort of pipeline as we've been experiencing it to get to maybe a more affordable or accessible in vivo treatment. But how would you speak to some of those questions that come up around access and equitability?
Yeah, no, that is, as I said, I think it's a bigger issue than the scientific issues. And the current approved gene therapies, like for sickle cell, for ALD, MLD, et cetera, they cost about $2 to $3 million for the cell product, and that's obviously not sustainable anywhere. Certainly not in low and intermediate income countries. And so it's going to take these technical breakthroughs. So if we can give a therapeutic gene therapy with LNPs by an intravenous injection, the cost to do that would be much, much less, both the hospital costs as well as the product costs, because you're not making individual cell products per patient. And that should be much more transportable. Like, you may need a cold chain like you would for a vaccine, but that may be all you need. You don't need a GMP facility. You don't need a bone marrow transplant unit. So I think those kind of advances, and especially in vivo is the great hope, should really bring down costs and make these things more widely available.
And in some ways, the global work that's been done around vaccine cold chains and being sure that that those life-saving interventions are available around the globe may have almost primed the pump a little bit for some of what you're describing.
Right, absolutely. They've described the pathway. I mean, there's even the clinic set up for distributing vaccines, et cetera, that in theory could do the same thing. And if the ideal would be that they're so safe that it's just basically an outpatient infusion of the drug, and they watch you for a half hour afterwards, just like giving immunoglobulin or whatever. Hopefully, it will become that routine, and that relatively easy to deliver. Right now, it's a month in the bone marrow transplant unit after the high dose chemotherapy, and just the clinical costs alone are probably 200 to $300,000 on top of the drug product. So again, hopefully, if we can simplify it down to in vivo delivery, but it's challenging. You have to ideally get it only to the stem cells or mostly to the stem cells at high efficiency.
So from your lips to God's ears, I mean, I really hope that we see an affordable intervention, a curative intervention world over. In the very last minute, because we have so many learners in our field and folks in training from around the globe, what words of inspiration, of guidance might you offer those who are here at the very beginnings of their careers with us today?
So I've been in science for about 40 years. It's a frightful number. And it's just be curious. Someone mentioned something in lab I haven't heard of. I want to know what it is. And so just, you build up your scientific knowledge base piece by piece and just stay with it. And something new that you haven't heard about comes up, don't be intimidated, learn about it and, incorporate it into what you do.
Well, thank you for that. And I'll just end with another word of inspiration as the chat that came in. One of your patients is here with us in the chat today, and they express such gratitude for your work and what you've done for their son. And I'll sort of echo that. And on behalf of the Jeffrey Modell Network and on behalf of immunologists world over, thank you for what you do. And to those who joined, thanks so much. So grateful that you're with us today, and I hope you enjoyed the webinar.
Thank you, Jordan.